Regarding drug receptor interactions, we know that our body is full of receptors everywhere, intracellularly, on the cell membrane, and even intranuclearly, inside the nucleus. But what we need to understand is that an unoccupied receptor does not really increase or decrease the intracellular processes. The average processes, the average activity of the intracellular processes will be considered as a baseline. However, so these receptors are, we consider them as signals detectors, while the drugs as molecules binding and searching and targeting those receptors are considered as signal initiators, and receptors are signal detectors. Upon binding to the receptors in a three-dimensional pattern, key and lock fitting between the drug and the receptor, if the drug is an agonist, means it is a stimulant to the receptor, it has the capability of activating that receptor in a way that it increases the intracellular activity, leading to certain biological responses. Now, whatever the intracellular activity that can be increased, there will be a second messenger. There will be a second messenger. So this drug is referred to as an agonist whenever it's able to bind in a three-dimensional key and lock fitting pattern. Then the drug is considered as an agonist as long as it can activate the intracellular processes, or the receptor action will be activated. The intracellular processes are considered the second messenger. The drug signaling the first messenger as a signal initiator, as a first messenger. The second messenger, or the effector molecules, are a group of cascade of events. Let's say there are many second messengers can be found intracellularly, like IP3, inositol triphosphate, or diacylglycerol, DAG, cyclic AMP when it is activated by the way of adenylyl cyclase enzyme. All these are considered as second messengers. Now, for the receptor as a macromolecule that is ready to bind with an agonist, here this is a diagram, an illustration that can shows you the agonist A, a competitive antagonist or an inhibitor, which is B, an allosteric agonist, which is C, and an allosteric antagonist or an inhibitor, which is D. So going back to the dose response curve, which is a graphical representation of the pharmacodynamic interactions. If we have the agonists alone, we can see that there is an active binding site. This one is the active binding site. The active binding site is the site where the agonist tries to bind to activate the intracellular messengers and produce the biological response. If there is another drug which is a competitive antagonist that has the same three-dimensional key and lock fitting pattern with the three dimensions of the receptor binding site, active binding site, then this drug is considered as a competitive antagonist or competitive inhibitor. So who will win at the end? The one with the higher concentration, as long as this one is a competitive inhibitor or antagonist, as long as it is a competitive. If it is a non-competitive or an irreversible antagonist, then the story will be different. The story will be that the agonist, whatever you increase the dose of the agonist, you will not be able to to stop the competitive inhibitor from blocking the receptor. But here, this is the S-shaped sigmoid diagram of the log dose response curve, which is a graded dose response curve for the drug A alone, if we use the agonist alone. But if we add the competitive antagonist, B, to the receptor before adding the agonist, we will notice that we will need a higher concentration, the dose, in order to achieve the same Emax or efficacy, the maximal response of drug A alone. So at the end, we will be able to achieve the efficacy of the agonist, of the full agonist, even in the presence of a competitive antagonist, but the only condition is that we need to increase the dose or increase the concentration at the receptor side. So the competition will be in favor, in that case, of that for the agonist, not for the antagonist, not for the competitive inhibitor. All right, so that's why the curve will take the same shape as that of the agonist alone, the same S-shaped, and the same Emax is achieved, but the curve is said to be, or is described to be, shifted to the right side of the original curve of the agonist alone. That is because of the presence of a competitive antagonist with the agonist competing with it, forcing us to increase the dose of the agonist in order to achieve the same Emax. All right. Now coming to the allosteric binding. Now we have mentioned about the active binding site. If you notice here, there is what is called an allosteric binding site. In the same receptor, macromolecule, there is an active binding site and there is an allosteric binding site. The allosteric binding site, if it is bound with an allosteric activator, actually what an allosteric activator will do is that it improves or enhances the binding of the agonist three-dimensionally to the receptor in a way that it can produce, the agonist will produce a higher than the 100% level of the efficacy that it can produce without that allosteric agonist. So allosteric agonist or activator, it produces better fitting three-dimensionally between the agonist and the receptor side, the active binding side, in a way that it can produce, the agonist will produce a higher Emax than without that allosteric agonist. Now, in the same way, there is what's called an allosteric inhibitor, and this allosteric inhibitor is capable of, when binding to the allosteric binding site, it is capable of distorting the three-dimensional fitting between the agonist and the receptor binding site. So the receptor binding site will not be easily recognized by the agonist in a key and log fitting pattern. So once they don't fit, the receptor will not be activated and the result will be A plus D. You see the efficacy has been reduced. This is the, no matter how much you increase the dose of the agonist, since the receptor conformational three-dimensional shape is changed because of that allosteric inhibitor or antagonist. We have examples for that. We're going to mention it later. But as long as the allosteric antagonist has altered the shape of the receptor binding site, active binding site, then the agonist will not be able to produce the same Emax or maximal effect. So the allosteric activator improves the three-dimensional fitting of the agonist with the active binding site, while the allosteric inhibitor or antagonist distorts the three-dimensional fitting in a way that impedes or interferes with the binding of the agonist. Now the receptor locations, as we mentioned, the body, organs, tissues are full of receptors, either on the cell membrane, such as the receptors that are G-protein coupled, GPCR, it's G-protein coupled receptors. Ligand-gated ion channels, all are present on the cell membrane. Enzyme-linked receptors, a receptor which is present like insulin receptor, which is a receptor that is available on the cell membrane, but it is linked intracellularly to an enzyme which is tyrosine kinase, such as in cases of insulin receptors. Calcium channels always are located on the cell membrane, and they are voltage-gated ion channels to which calcium channel blockers can bind and block the influx of calcium from the extracellular to the intracellular part. Other receptors can be intracellular and inside the cytoplasm, such as the cyclooxygenase enzyme, COX enzyme, or the angiotensin converting enzymes, which are responsible for the activation of angiotensin 1 into active vasoactive angiotensin 2 vasopressor agent, and to which the ACE inhibitors act on. These are all targets, drug targets. We said that receptors are drug targets, while drugs are signal initiators. All right. The other examples are of intranuclear receptors inside the DNA, such as antiviral drugs and some of the anti-cancer drugs. They are actually targeting the DNA of the neoplastic cells with high specificity or selectivity towards the neoplastic cells. Yes, and the glucocorticoid receptors are also intracellularly. So these are the receptor locations and the receptor binding sites. These are the examples of macromolecular receptors, such as enzymes, nucleic acids, which is the DNA or RNA, structural proteins, such as in cases of paclitaxel, which is an antineoplastic drug targeting the tubulin, which is one of the structural protein. Trimethoprim is an antimicrobial agent that targets the enzyme, which is important for the synthesis of the bacterial pyrimidine. Which by inhibiting the enzyme dihydrofolate reductase, so it targets that enzyme, and then it reduces the synthesis of an important element for the synthesis of DNA, which is bacterial DNA, which is pyrimidine. Erythromycin, which is a macrolide, which targets the protein synthesis by inhibiting the 50th subunit of the bacterial ribosome, inhibiting the bacterial ribosomal growth, and therefore ultimately the bacterial cells will be allowed to be attacked by the human immune system.